The DNAse test, determining the production of deoxyribonuclease enzymes. DNAse, or deoxyribonucleases, are enzymes produced by bacteria that are excreted into the environment in order to break down DNA as a nutrient source. The DNAse test is a very simple and easy test to run. You begin by drawing a line down the center of your plate, and then labeling each half of the plate with the bacteria that you will be inoculating them with. For this test, we will be using Staphylococcus aureus and Enterobacter aerogenes. The transfer of bacteria from the slant to the plate is completed using aseptic technique. The agar will not be stabbed or punctured. It will be a simple straight line plate inoculation. Using a straight line during the inoculation will make your results much easier to read after incubation. Be sure to sterilize your loop in between each inoculation. Once the inoculation is completed, you will place the plate into the incubator until your next lab period. It is important to label the plate correctly and make sure that the side labeled Staph aureus is what is inoculated with Staph aureus. Shown here is the simple straight line inoculation to be used. Do not puncture or gouge the agar. It is not necessary and can make results more difficult to interpret later on. You will be reading the results of this test using the reagent hydrochloric acid. Hydrochloric acid will cause any remaining DNA in the plate to precipitate. Here, HCl is being used to flood the top of the plate. As the HCl, or hydrochloric acid, absorbs into the agar, the DNA that is left behind will precipitate and cause the agar to become cloudy. If bacteria have produced DNases, there will be no DNA to precipitate. This will cause a ring or halo to form around the bacteria after the addition of hydrochloric acid. Here on the right, this organism is positive for the production of DNases. On the left, this organism is negative. The DNase test is used oftentimes in identification of pathogenic Staphylococcus. The production of DNases has been linked to the degradation of biofilms. This is thought to enhance an organism's ability to invade a host or to spread from a site of origin to increase infection throughout the host. The DNase test is a very simple test that is used in determining the presence of DNase production.